Across multiple cancer types, there has been countless efforts over the years to try and come up with new early detection and screening biomarkers. The goal is that this technology and this approach would lead to a blood test for early detection of cancer, especially in particular cancer types. We know that the earlier we can detect a cancer, the more likely we are able to achieve cure. So if you take a blood sample and you spin it down and remove all the cells, we've known for a couple of decades now that there are fragments of DNA that are floating around in the cell-free part of blood called plasma. If you've taken this blood sample from a pregnant mother, then a fraction of this DNA is contributed by the fetus. If you've taken this sample from a patient with cancer, a small fraction of these fragments is contributed by the tumor, and you can detect these fragments. The modal length is about 167 base pair fragments. And that number is significant because it's about the same length of DNA that you would find wrapped around in a single nucleosome. And you can tell that they're tumor specific because they would oftentimes carry the same mutations you would otherwise find in the cancer's DNA. And so could we exploit these differences in fragmentation patterns to tell which plasma sample is from a patient with cancer versus which one's from a healthy individual? We came up with a list of 10 different features that seem to be informative. When we look at all of these features put together, each one of them by itself somewhat informs our ability to tell patients from cancer versus healthy individuals. But when we combine them together in a machine learning model, our ability or our accuracy of detecting a sample with cancer versus a healthy individual becomes greater than 90%. Unlike sort of traditional measures that depend on mutations and other methylation patterns and so on that measure tumor fraction, measuring this fraction of aberrant fragments is actually very straightforward. We're talking about picograms of DNA, which you could effectively get from very small amounts of blood sample. You don't need a lot of DNA, you don't need a lot of sequencing depth, and it sort of could be very cost effective. So what's made this research possible are advances in two aspects. One set of advances is in genomic sequencing technologies. Genomic sequencing has become much more cost-effective and accessible than it used to be 10 years ago. And the other piece that has made it possible are these advances or our ability to use computational tools and machine learning models to try and interrogate the features we find in sequencing data and really combine them and integrate them. On one end, there is the definitive diagnosis using a tumor biopsy, after which you can initiate cancer therapy based on stage and tumor type. On the other hand, you have individuals who may be potentially at average risk of cancer, a population-wide risk of cancer, and you're interested in screening for cancer types that are very common. Where I see some of this technology being applied is somewhere in the middle in this space that's developing called early detection. And so we're considering the use of this technology in patients who are at higher than average risk. They either have some sort of presentation of sort of non-specific symptoms where it's not clear whether the patient has cancer. I'm very excited about what this means for access to early detection testing and what this could mean for both patients with limited resources in the US and globally, and how this could potentially change how we do early detection across the world.